Aloha and welcome to Botany 101. My name is Angela Nishimoto. If you recall, last time we talked about water in plants. It was a challenging chapter, right? We talked about diffusion, we talked about osmosis, we talked about active transport, right? And we also talked about movement of water in xylem and also movement of solutes in the phloem, the food, or photosynthate, right? So we talked about those topics, and we also got into a prelude for photosynthesis. We talked about some components of photosynthesis, right, including carbon dioxide, including water, right, including light, and then we talked about chlorophylls, okay? Today we're going to continue that discussion and get into photosynthesis, get into respiration also. So today's lecture is going to be very challenging. It's going to be kind of hard to understand, I think, unless you've been through it before. So let me encourage you right now, okay, to please contact me if you have any questions about what we're covering, okay? So please, phone me, email me, come and see me in my office, okay? If you can't make my office hours, you can make an appointment, okay? So today we're covering plant metabolism, Part 2, okay? So our outline of topics, we're going to hopefully talk about all of these. Photosynthesis, cellular respiration, a kind of respiration known as fermentation, and we're going to talk about secondary metabolites, okay? Now photosynthesis, okay, takes place in the chloroplast in the cell, the plant cell, okay? And photosynthesis takes place in two parts. You have the light reactions that happen in the grana, and you have the Calvin cycle that happens in the stroma, okay? So the light reactions happen in the grana, okay? Remember what grana are? They're stacks of thylakoids, right? Making up a granum, okay? So you have the stacks of thylakoids in which you have these photosystems embedded. We talked a little bit about photosystems last time, okay? So I'll remind you that photosystems are an array of photosynthetic units, and photosynthetic units are made up of pigment molecules, okay, 200 to 400, something like that, all right? So in the light reactions, okay, a photon, which is a packet of light energy. Remember that light is both a wave and also a discrete unit, okay, known as a particle, okay? And this particle is known as a photon. So the photon hits a chlorophyll molecule, and that splits water, okay? The water releases electrons and hydrogen ions, also known as protons, because if you remember, hydrogen atoms are a proton and an electron, and if it loses the electron or donates the electron, it becomes a proton or a hydrogen ion, okay? Remember that hydrogen ions cannot pass through the membrane. Only oxygen gas, carbon dioxide gas, and water can freely pass through a membrane, okay? Another thing that happens that is it releases oxygen gas, okay? So the electrons and the hydrogen ions and the oxygen gas comes from the split water molecule, okay? Now then, electrons then pass through an electron transport system. Okay, so first of all, let's talk about the first part. Here's water. Okay, water is split by light, okay, giving off oxygen gas, releasing hydrogen ions, and also electrons. Okay, this all comes from water. Oxygen gas, hydrogen ions, and electrons, okay? Now then, the electrons are passed along an electron transport system, okay? And the electrons, of course, come from the split water molecules, okay? So here's the electrons. They're actually passed from an electron acceptor, from something we'll talk about later, P680, okay, to electron acceptors, and other electron acceptors, other electron acceptors, etc. Okay? Now then ATP is produced, Okay, show you that. Okay, what happens is that the hydrogen ions, all right, are actually on this side of the membrane. They actually are transported from one side of the membrane from lower concentration to the other side of the membrane at higher concentration. Okay, because they're at higher concentration, they will diffuse back through an ATPase or ATP synthase, thereby generating ATP from ADP, adenosine diphosphate plus a phosphate group, to make ATP, adenosine triphosphate, which we talked about last time. Okay. Another thing that happens is that some of the hydrogens from the split water molecule is used to form reduced NADPH, nicotinamide adenine dinucleotide phosphate. That's reduced NADP. Okay, so you can remember NADPH, think of the P as standing for photosynthesis. Okay, because later we're going to be talking about NADH, okay, in respiration. All right, so NADPH is the electron carrier in photosynthesis. Okay, so here you have the electrons finally being passed to NADP plus the hydrogen, forming NADPH. Now the second part of the photosynthesis happens in the stroma, and it's called the Calvin cycle. Okay, so the product of the light reactions may proceed, along different pathways depending on the type of plants, but they will all go through the Calvin cycle. Okay, the Calvin cycle actually uses ATP and NADPH energy and electrons. Okay, so energy from ATP and electrons from NADPH, okay, which we just talked about. Okay, and during the Calvin cycle, this is when carbon dioxide comes in. Okay, so the carbon dioxide is combined with a sugar, a 5-carbon sugar known as ribulose bisphosphate, also called RUBP. All right, it's a 5-carbon sugar, right? And this complex is then converted to sugars, okay, like glucose. Okay, so here we have RUBP, okay, combined with carbon dioxide. Let's see, here, RUBP combined with carbon dioxide, okay, to eventually form sugars. Okay. Now some of these sugars are recycled, okay, others are stored as starch. Okay. So this 3-phosphoglyceric acid, or 3-PGA, okay, is the first stable compound from carbon dioxide and RUBP. Okay, so 2 molecules of 3-PGA are formed. Okay, and here it is. Okay. Now some grasses, tropical grasses and dry area plants fix carbon differently, and we'll be talking about that a little bit later. Okay, now we're going to talk about the history of the discoveries of the various parts of photosynthesis. So in 1772, Joseph Priestley won a medal. He actually demonstrated that mint plant restored, so-called restored oxygen gas, so a mouse could live in a bottle. Okay, so the mint actually produced oxygen gas. So the mint restored oxygen so the mouse could live, because of course the mouse would have been breathing. Okay, 6 years later in 1778, young Ingenhouse, okay, showed that the air was restored only when the mint got sunlight. Okay, so sunlight was necessary for this oxygen to be restored. Okay. 4 years later, Jean Senebier, okay, showed that plants needed carbon dioxide. Okay. Now 4 years later, in 1796, Jan Ingenhausen, once again, he was among one of the discoverers, okay? He was the one to discover that carbon is a nutrient for plants, that plants need carbon, okay? And got it from carbon dioxide, right? And then 8 years later, Nicolas Theodore de Saussure actually discovered that water is involved. So de Saussure actually showed that water was involved, which was the last piece of the puzzle for photosynthesis to actually be delineated. Okay? Now we're going to look again at the light reactions in greater detail. Remember, they happen in the grana of the chloroplast. Okay? So in 1905, Albert Einstein, okay, discovered, or he actually stated, that the photoelectric effect came from a photon, the actions of a photon. Okay, so what is a photoelectric effect? Okay, in metals, they become positively charged. Okay, because radiation forces out electrons. Okay, and what that does, it depends upon the wavelength of the radiation reaching it. Okay, so it actually becomes positively charged. That's called the photoelectric effect. Okay? And in 1921, he got the Nobel Prize for this work. Okay, remember we talked about photons a little bit earlier. Recall that chlorophylls absorb in the violet to blue and also the orange-red to red, right? And it reflects green. That's why leaves look green to us. Okay? Now, this was actually demonstrated ingeniously in 1882 by T.W. Engelmann. Okay, he performed something called Engelmann's experiment. Okay, what he did is he got one strand of a filamentous alga known as Spirogyra, one strand of alga. It's a freshwater alga, okay, a green alga. And what he did is he put it on a slide. Put it on a slide with the cover slip and mounted it there in water, in fresh water, and he shined light through a prism, okay? Of course, when light is shined through a prism, white light breaks up into its component parts from red to orange to yellow to green to blue to violet, okay? He put in there, in the water, oxygen-loving bacteria, okay? So as the light was shining on this alga, okay, the oxygen-loving bacteria were congregating toward the blue area and toward the red area of where the spectra was. Okay, so that demonstrated that oxygen was evolved, okay, during photosynthesis, and that photosynthesis happened in the blue, violet, and in the orange-red. So that was kind of an interesting experiment. It's like what they call an elegant experiment when you're using very simple tools to support some very important hypotheses. Okay. So we have these things called photosystems, which we talked about a little bit. Okay, there are two photosystems. Okay, so photosystems are arrays of photosynthetic units. Photosynthetic units are made up of pigment molecules. Okay, about 200 to 400 of them. So first of all, we have photosystem 2, okay, with a reaction center molecule of chlorophyll A. It's called P680. Okay, P stands for pigment. 680 stands for the wavelength at which it actually absorbs the light, okay, at 680 nanometers. Okay, so photosystem 2 is the first photosystem, and then we have photosystem 1. Okay, photosystem 1 and photosystem 2 are actually named in the order in which they were discovered, but in the process of photosynthesis, photosystem 2 comes first. Okay, now photosystem 1 has a reaction center chlorophyll that has P700, or P stands for pigment again, and 700 nanometers at which it absorbs light. You see a little bit different between photosystem 2 and photosystem 1. Okay? This is showing photosystem 2. okay, with the different kinds of pigment molecules in it. Okay, P680 is a reaction center chlorophyll. Okay, and here's photosystem 1. Okay, with pigment molecules, and P700, the reaction center molecule, the reaction center chlorophyll. Okay. Now, photosystem 2 is also known as a water splitting photosystem. Okay. So the photons, okay, are actually absorbed by pigment molecules, which ricochet it around until it reaches P680. Okay. Then electrons get excited. Okay, so that means that they're at a high energy state. So excited electrons are unstable, right? They go back to their ground state fairly quickly, but for now they're boosted to a higher energy level, and then they're passed on to electron acceptors. Okay? And then what that does, it actually splits water too. Okay, and water then gives up oxygen gas, hydrogen ions, and electrons, like we talked about earlier. Okay? So here we go. Okay, water here is split by light from here. Okay, giving up water, 4 electrons, which go through the electron transport system, and 4 hydrogen ions, or 4 protons. Okay. Now, electron flow and photophosphorylation. The electrons are passed down an electron transport system. Okay, so these excited electrons are passed to electron acceptors like phaeophytin, then to plastoquinone, then to the cytochromes. Okay. Now the hydrogen ions are moved across the membrane like we talked about a little bit earlier. Okay. And ATP is formed from ADP plus a phosphate group. It's phosphorylated. Okay, so ADP, or adenosine diphosphate, is phosphorylated to form adenosine triphosphate, or ATP. And that's done by ATP synthase. Okay, ATP synthase is a membrane protein complex. Okay. So electrons are passed down an electron transport system. Okay. Hydrogen ions are moved across the membrane using the energy from the electrons, which goes from a higher excited state and gradually releases energy and goes down to a lower state. And that energy is used to pump protons across the membrane, okay, into the thylakoid lumen, or the inner membrane space. I mean, not the inner membrane space, I'm sorry. But inside the thylakoid membrane, inside the thylakoid. If you remember, thylakoids are like coins, stacks of coins of membranes, right? And then what happens is that the hydrogen ions then diffuse back to the ATP synthase, producing ATP, okay, from ADP plus a phosphate group. Okay, now the NADPH generating photosystem is photosystem 1. Remember, photosystem 2 comes before photosystem 1, and photosystem 2 is actually the water splitting photosystem. So photosystem 1 is the NADPH generating photosystem. Okay? In the NADPH generating photosystem, or photosystem 1, the photons are absorbed, okay, and they kind of ricochet from one pigment molecule to another until it reaches P700, where it's absorbed. The electrons are excited, and then they're transferred to electron acceptors. The first one is iron-sulfur acceptor molecule. Then it goes to ferredoxin, okay. Then it goes to FAD, okay, which then is used to reduce NADP, okay. And that reduces NADP to NADPH. Okay, so we have an overall movement of electrons, okay, from water to photosystem 2 to photosystem 1 to NADP. Okay, so it goes from water to photosystem 2 to photosystem 1, right? Then to NADP. Okay, so that's how electrons flow. Okay, so here water gives up electrons, go from photosystem 2 to photosystem 1 that actually end up reducing NADP. Now we have chemiosmosis. Okay, so chemiosmosis actually happens inside the thylakoid membrane. Remember that water is split, producing what? Do you remember what water split produces? Okay, think about it for a few seconds. Okay, it produces oxygen gas, hydrogen ions, and electrons, right? Okay, remember? Water is split, producing oxygen, hydrogen ions, and electrons. Okay? Then electrons go through an electron transport system, okay, like we just kind of showed through the illustration. The hydrogen ions move to the inside of the thylakoid in the lumen, and the hydrogen ions accumulate, okay, forming a proton gradient. Then the hydrogen ions move through the protein complex known as an ATPase, and that produces ATP. Okay, so we have seen how the light reactions produce both ATP and also NADPH. Now both of these molecules are important in the second part of photosynthesis known as the Calvin cycle. Okay, so both ATP and NADPH are produced in the light reactions and they're used in the Calvin cycle. Okay, so anyway, so let's kind of take a look at that. Okay, so we have the electrons going from one electron acceptor to the electron transport system. Okay, hydrogens move to the inside of the thylakoid in the thylakoid lumen, right? And they accumulate here until they have a high concentration, okay, of protons, right? Then they'll diffuse back to this ATPase and produce ATP. Now the Calvin cycle, we're going to look a little bit more closely at it. We're going to re-examine it. Anyway, so the Calvin cycle happens in the stroma, which is the jelly-like liquid inside the chloroplast, because remember, all of this happens in the chloroplast, one of the organelles inside of the plant cell, in the leaves especially. Okay? So Melvin Calvin studied these reactions of the Calvin cycle using green algae and radioactive carbon, okay, as the algae went through reactions. Okay, so carbon dioxide is fixed to form carbohydrates. Okay, the carbohydrates that are produced, okay, helps to facilitate growth. It helps the development of roots, stem, leaves, flowers, other structures of the plants. And this is very important because this is the main way that carbon enters the web of life. Okay, all right, very important. So carbon dioxide is fixed to form carbohydrates. Okay, it's the main way that carbon enters the web of light. Okay, so then happens during the Calvin cycle. 6 carbon dioxides join with 6 RUBPs, ribulose bisphosphate, the 5-carbon sugar. Okay, and that's actually facilitated with the help from Rubisco, which is an enzyme. All right, so Rubisco is actually called that's a short, an acronym for ribulose bisphosphate carboxylate oxygenase. Okay, so both carbon dioxide and oxygen can be substrates for Rubisco. Okay, so we'll talk about that a little bit later. Okay, so here's Rubisco. Okay, here's carbon dioxide joining with the RUBP. Okay. Now the 6 carbon complexes then split into 12 3-carbon, 3-phosphoglyceric acids, or 3 PGAs. Okay? So the first is the first stable compound that's formed during photosynthesis, 3 PGA. Right? Now then, NADPH and ATP then give energy and electrons to reduce this phosphoglyceric acid. Okay? So it reduces the phosphoglyceric acid and eventually becomes, okay, so here's 10 glyceraldehyde 3-phosphates. I think I must have skipped over something. It's 10 glyceraldehyde 3-phosphates, right? So it actually transforms phosphoglyceric acid into glyceraldehyde 3-phosphate. Okay? Now 10 of these glyceraldehyde 3-phosphates then join to 6 ATP to form 6, okay, ribulose bisphosphates, or RUBPs, that 5-carbon sugar, right, that we started the cycle with. Okay? So there are 2 glyceraldehyde 3-phosphates that are netted, okay, through the Calvin cycle's turns. All right? So 12 molecules. Okay? So that regenerates RUBP, and then this GA3P, okay, is used to form glucose or starch or cellulose, okay, or lipids or amino acids. Okay? So this 3-carbon compound eventually goes off to do different kinds of metabolic pathways to actually form those compounds. Okay? Now the plants that do this kind of photosynthesis is known as, or Calvin cycle, is known as the C3 plants, okay, because rubisco forms a 3-carbon, 3-phosphoglyceric acid, or 3 PGA. Okay, so it's called C3 plants, since rubisco forms a 3-carbon compound. Okay? Now rubisco, the enzyme, which is the most prolific enzyme in the world, there's actually more rubisco than any other enzyme in the world. Think about it, all these plants have rubisco, okay? So rubisco can fix both carbon dioxide and oxygen gas, which we talked about a little bit earlier, okay? So when it's acting as an oxygenase, okay, it's actually acting to use oxygen as a substrate, that can lead to photorespiration, which we've talked about before. Okay? Okay? So rubisco joins with oxygen gas and releases carbon dioxide. Okay, so it actually joins with oxygen and releases carbon dioxide, which is the opposite from photosynthesis, right? But this is not as wasteful as it may seem. Okay, it allows the C3 plants to survive heat and dryness. Okay, helps to dissipate ATP and accumulated electrons, and also prevents photooxidative damage. Remember we talked about that last time, right, with the C3 plants? Okay, but no ATP is produced. In fact, ATP is actually consumed in this process. Okay, so here is photorespiration. So if the plant is actually going through photorespiration, the RUBP joins with an oxygen, okay, helped by rubisco. Okay, and it actually gives off carbon dioxide and actually uses up an ATP. Okay, so it is a little bit wasteful, but it also does kind of help prevent photooxidative damage, as we just said. Now there's another kind of photosynthesis known as C4 photosynthesis that happens in sugarcane, corn, and sorghum, and at least 1,000 other species of tropical grasses, and also dry area plants. Okay, all these plants have in common is they all share Crohn's anatomy, okay, in the leaf. Okay, what is that? Okay, that means that they have 2 kinds of chloroplasts. Okay, there are chloroplasts in the bundle sheath that surrounds the vascular tissue. Okay, that's here. Okay, and these chloroplasts are large. They have lots of starch grains in them, numerous starch grains. Okay, they also have poorly developed grana. Remember grana, that's where the light reactions happen, right? And then you have other kinds of chloroplasts in the mesophyll cells. So here's the mesophyll cells, and these chloroplasts are small, okay? They're small, and they have well-developed grana, okay? So the light reactions happen a lot in the mesophyll cells, of course, okay? Now what C4 photosynthesis does, it produces a 4-carbon oxaloacetic acid, so C4, 4-carbon, right? So what happens is that there's a 3-carbon phosphoenolpyruvate, or PEP, okay, combines with carbon dioxide with the help of PEP carboxylase, and you notice that the name is PEP carboxylase. It means that carbon dioxide is the substrate for this particular enzyme, okay, carbon dioxide only, okay. So the carbon dioxide, okay, is transported to the bundle sheath cells where the carbon dioxide enters a normal Calvin cycle, like in the C3 plants, okay. Now other features of C4 plants, so let's look at this first. Okay, so here you have the bundle sheath cell and the mesophyll cell. Okay, so here's PEP, okay, phosphoenolpyruvate, okay, joining with a phosphate in the mesophyll cell to produce malate, which is an acid, okay, and in the bundle sheath cell, it gives off carbon dioxide, and then actually then goes through the Calvin cycle, okay. Now, other features that C4 plants have, okay, is that they have high concentrations of PEP carboxylase, of course. So that allows them to function at higher temperatures, okay. That allows the C4 plants to actually thrive under conditions that are too hot for C3 plants. Okay, so when it's warm like that, that's actually optimal conditions for C4 plants. Okay, when it's kind of hot and dry. Okay, another thing that this does is the C4 photosynthesis allows for a major reduction of photorespiration. So photorespiration doesn't happen very much in C4 plants. Okay. Now we also have another kind of photosynthesis known as CAM photosynthesis. CAM stands for crassulation acid metabolism because it was first discovered in crassulas, a kind of succulent. So it happens in cacti, orchids, bromeliads, and succulents. Okay, so these plants are often stressed by limited water and high sunlight. Okay, now some succulents do not do CAM photosynthesis, and some non-succulents do. Okay. Now, so generally these plants have no well-defined palisade cells, okay? But these 4-carbon compounds accumulate at night, mostly malic acid, which is PEP plus carbon dioxide with help from PEP carboxylase, okay? And these acids break down during the day, okay? Releasing carbon dioxide to be converted to carbohydrates, all right? And the stomata are closed during the daytime to conserve water, okay? So this allows the plant to function well under limited water resources and a lot of light, okay? So you actually have a separation in terms of time for CAM photosynthesis, whereas in C4 photosynthesis you have a separation in location, between the bundle sheath and the mesophyll cells. Okay, so during the night, carbon dioxide come in. This is when the stomates are open. Okay, so PEP is actually joined to carbon dioxide to form oxaloacetate that forms malate. Okay, and that's stored in the vacuole of the plant. So here you see the vacuole swollen with malate or malic acid, okay, and water. Okay. Now during the daytime, the stomach closes. Okay. Then what happens is that the malate actually leaves the vacuole, okay, and goes back to oxaloacetic acid, goes back to PEP, okay, and then is used, okay, to do the Calvin cycle. Okay. Now we're going to switch gears now and go from photosynthesis to respiration. So if you recall, photosynthesis is an anabolic process, right? It actually builds up larger molecules from smaller molecules. Now respiration, so generally when photosynthesis happens, okay, you actually build molecules, you build sugars, you build starches, you build cellulose, you build proteins, you build fats, okay? So the solar energy that's collected by the plants is sometimes stored in wood, and coal and oil are actually made from photosynthesis from eons ago, you know, many centuries ago, right? So actually if you burn wood or coal and oil, you know, you'll have a big conflagration, right? You have a big release of energy in the form of heat and light, and not much usable energy is actually available to be used to do any kind of work, okay? That's like an explosive release of energy. So cellular respiration is something that allows the release of energy in small, controlled steps, controlled by enzymes, okay? So respiration is actually the burning of compounds in small steps, okay, releasing energy in the form of ATP, okay? So this is actually looking at the pathways for glycolysis, which happens in the cytoplasm, okay? So the essence of respiration is actually the release of energy from glucose by oxygen, okay, for aerobic respiration. Energy is released, and carbon dioxide and water are byproducts, okay? So it's actually broken down to carbon dioxide, okay? It occurs 24/7 in all living cells, whether or not photosynthesis is going on at the same time in the same cells, okay? So cellular respiration happens in all cells all the time, okay? It starts in the cytoplasm and ends in the mitochondrion for aerobic cellular respiration. Okay, so the energy in chemical bonds is released in small, controlled steps. So aerobic respiration, you need oxygen gas. Okay, so the equation is, you know, you have C6H12O6 or glucose broken down by 6 oxygen molecules. So sugar is partly broken down by oxygen gas, or sugar is broken down partly by oxygen gas to release carbon dioxide and water and energy in the form of ATP. Okay? So this actually shows a cycle. Okay, so photosynthesis actually uses carbon dioxide and water, okay, uses sunlight energy to actually convert carbon dioxide and water to more plant material, okay, and also different kind of metabolites, different kind of organic compounds, you know, carbohydrates, lipids, nucleic acids, and proteins, okay. So then what happens, it also generates oxygen gas, right? So then you have animals such as this woman, and also fungi during cellular respiration, plants do cellular respiration too, okay. So what they'll do is they'll actually use organic compounds, especially in the form of sugar, and break it down with oxygen, and then use it to release energy in the form of ATP, which is used to power all work in the cell, okay, whether it's animal cell or plant cell. Then it releases water and carbon dioxide, okay? Now, anaerobic respiration and fermentation, which are other pathways, a little bit slightly different. They actually do go through glycolysis, but they actually happen without oxygen, but oxygen doesn't come in until later. Okay, so this is the chemical recipe or chemical equation for the production of ethanol. So glucose is converted to ethanol and carbon dioxide plus energy in the form of ATP, ethanol or ethyl alcohol. Okay, this equation is when glucose is converted to lactic acid, okay, plus energy in the form of ATP. Okay, so these forms are still done by some bacteria and other organisms, okay, when oxygen is unavailable. So here's this nice graphic here, okay, where you have 2 ATP invested, glucose is split and becomes pyruvic acid eventually after many transformations, okay. 2 ATP are netted whether it's lactic acid fermentation or alcoholic fermentation, producing ethanol, okay. Now back to aerobic respiration. Okay, so the first part, glycolysis, needs no oxygen gas. Okay, so aerobic respiration happens in 3 parts. You have glycolysis that happens in the cytoplasm. Okay, you have the Krebs cycle that happens in the mitochondrial matrix, the matrix of the mitochondria in the center of the mitochondrion. And then you have the electron transport that happens on the inner membrane. Remember, the inner membrane of the mitochondrion is infolded. There's a lot of folds in it, giving it a lot of surface areas. So there's a lot of electron transport systems on it. Okay? So glycolysis happens in the cytoplasm. Okay? So the first part needs no oxygen, and there are 3 main steps or several smaller steps, each controlled by enzymes. Okay? First of all, glucose is converted to fructose with 2 phosphates. Okay. Then what happens is that fructose then splits into 2 3-carbon glyceraldehyde 3 phosphates. Okay. Then hydrogen, energy, and water is removed. Okay. And it results in the formation of pyruvic acid. So there's some small amount of energy that's released and hydrogen's removed. Okay, so there are 2 ATP that are needed to start the process. So by the time pyruvic acid actually is formed, okay, you will have 4 ATP produced from the energy released during the process. So you net 2 ATP during glycolysis. Okay, so the hydrogen ions and electrons are picked up by NAD to form NADH. Remember I said NADPH is in photosynthesis and NADH is in respiration. Okay, there's still lots of energy still left in the pyruvic acid. Okay, so here's glycolysis. You see ATP is invested here. Okay, it's actually transformed to a fructose. The fructose is eventually split, okay, into a glyceraldehyde 3 phosphate. Okay, then you have a ATP actually being produced here and here. Okay, so 2 ATP is actually used, all right? Then you have 4 ATP produced, okay, plus NADH, all right? So you have a net 2 ATP, so you minus 2 ATP plus 4 ATP, you have a net gain of 2 ATP. Okay? Now the citric acid cycle or the Krebs cycle, I call it the Krebs cycle. That's how I learned it, and that's how I'll teach it. Okay? So the Krebs cycle happens in the matrix of the mitochondrion, the very most inner part of the mitochondrion. Okay? It's named after Hans Krebs, the person who delineated the steps during the 1950s. He won a Nobel Prize in 1953. Okay? So before the Krebs cycle, the carbon dioxide is released from pyruvic acid. Okay, it's restructured to form 2-carbon acetyl groups, and that combines with something called coenzyme A. Okay, and that's called acetyl coenzyme A. Okay, that happens before it enters the Krebs cycle. Okay, it would have been formed before this. Okay, so then acetyl coenzyme A actually enters the Krebs cycle. Now, during the Krebs cycle, electrons and hydrogens are removed. Okay. Ultimately forming NADH and a substance called FADH2, which is flavin adenine dinucleotide or reduced flavin adenine dinucleotide. Okay, it's another electron acceptor. So NADH and FADH2 are just electron carriers. Okay, so don't get too scared about the acronyms and all the words. Okay, ATP is formed. Carbon dioxides are removed. So during the Krebs cycle, you have the completed breakdown of what was once the glucose molecule. Okay, you have the complete breakdown of what was originally the glucose molecule during the Krebs cycle. Okay, see here, you see this? Okay, so you start off with oxaloacetate, okay, joining with the acetyl group of acetyl coenzyme A. Okay, you have a 6-carbon substance, you have a 5-carbon substance, and you have 4 carbons with the carbon dioxides being given off, losing carbons. So it regenerates oxaloacetate at the end of one cycle. Okay. Producing ATP, NADH, and FADH2. Okay, now electron transport. Okay, it happens in the mitochondrial inner membrane. Okay, so NADH and FADH2 donate electrons to a series of carrier molecules. Okay, down an energy gradient. Okay. Ultimately, the electrons fall to oxygen. Remember that oxygen is among the most electronegative of all the elements, right? So the protons are pumped, okay, forming electrochemical gradient. Okay, so the carriers accept the hydrogen ions and release them to the intermembrane space, the space between the inner membrane and the outer membrane. Okay. And or other proteins, the ATPases, allow the hydrogen ions back into the matrix of the mitochondrion, and that forms ATP through phosphorylation. Okay, so the aerobic net ATP count is 36 ATP, okay, from one glucose molecule. Okay. So by the time it's over, the energy in glucose is converted to ATP. And of course, ATP energy is used to make parts of cells. It's used to make organs of the plant. It's used in active transport. Okay, and some of it is used to form other structures and allow certain kind of chemical reactions to go forward. Okay. Now, aerobic respiration. Right, so you see here the acetyl coenzyme A coming in here. Okay, joining with I'm looking for the oxaloacetate. Let's see. Here it is, oxaloacetate. Okay, forming a 6-carbon structure. Okay, actually then forms another 6-carbon structure, and then another 6-carbon structure, and then a 5-carbon structure after it loses the carbon dioxide. Okay, then it loses another carbon dioxide to form a 4-carbon structure. Okay, and then another 4-carbon structure, and eventually regenerating oxaloacetate. Okay. Now, anaerobic respiration and fermentation, another look at it. Okay, so in anaerobic respiration and fermentation, you have glycolysis only, and it occurs in some living things. Okay, so in these things, there are not enough oxygen to complete aerobic pathway. Okay, so the hydrogen is transferred back to pyruvic acid, which becomes ethanol or lactic acid, like we saw a little bit earlier. So you have a net gain of 2 ATP compared with 36 of aerobic respiration. So living cells can live with only so much alcohol around them, right? So when you have yeast fermenting carbohydrates to make beer or wine, okay, generally what'll happen is it'll keep on fermenting it, creating alcohol until it reaches about 12%. Once it gets to about 12% or over 12%, the yeast organisms begin to die. That's why wine is usually about 12% alcohol or 24 proof. Okay. So here's looking again at fermentation, alcoholic fermentation. Okay, we have a 3-carbon compound becoming a 2-carbon compound after giving up a carbon, producing ethanol. Okay, lactic acid fermentation. So this actually is part. So they undergo glycolysis first to net 2 ATP. Okay. So these anaerobic pathways, lactic acid fermentation and alcoholic fermentation, allow for the regeneration of NADH. Okay, so you can go back to glycolysis. But like I said, generally fermentation allows for only the production of 2 ATP as compared to 6 during aerobic respiration. Okay, now some factors that affect the rate of respiration. Okay, temperature is a major role in respiration. Okay, the higher the temperature, the faster the respiration rate. Okay, when the temperature goes from 68 degrees Fahrenheit to 86 degrees Fahrenheit, the respiration of plants will actually double or triple. Okay, the faster respiration happens, the faster the energy is released. That can lead to a decrease in weight. Okay, and the weight loss is usually offset with more photosynthesis, producing more sugar. Okay, now in harvested fruits and vegetables and seeds, respiration continues without sugar replacement. Okay, so what happens is the respiring cells convert stored energy to ATP, but only a little bit. Most of it is actually lost through heat. Okay, so they lose through energy through heat. So that may be one reason why in the grocery store you see the guy hosing down the fresh produce, right, to cool it down, as well as to try to restore some water to it because they lose water. It's kind of showing that with increasing temperatures, okay, growth rate can increase up to a certain point, okay, and then after a certain point you get problems and it decreases. Okay, here's respiration, okay, which stops. So fresh foods are refrigerated, okay, because under refrigeration you have a lower respiration rate, of course, because heat tends to accelerate the actions of enzymes up to a certain point, okay. It also retards the loss of water. It also dissipates heat. It also prevents the growth of mold and bacteria that destroy foods. Okay. Now another factor that affects the rate of respiration is oxygen. Plants need oxygen, of course, to do cellular respiration. Okay. So in flood, if you have a plant, okay, that has its roots soaking in water, okay, that will decrease the oxygen supply that's available to the roots, right? So what can happen is that respiration can decline, growth will decline, okay, the roots can rot, and maybe the plant can die. Okay, this is actually showing, looks like a plant in kind of dry area, but the same thing can happen because if the roots rot, the plant can't take up any water anymore. So sometimes when you're actually watering plants, okay, and you put too much water in there and they're sitting in water, what will actually happen is that the roots will rot, roots rot away, the roots die, and then the plant can't take up any water anymore. So the upper part of the plant looks like it's drying out. So then you, of course, think, oh, I should give it more water, you know, and that is actually the sure way to kill it. That's why sometimes when you overwater a plant, that's actually worse than not watering it enough, okay? So what they do is they commercially reduce oxygen levels, okay, to as little as 1 to 3%. Okay. Now it's not reduced to below 1% because that can result in an increase in fermentation, and of course we don't want that. So what you have is you have foods kind of stored in commercial warehouses, maintaining low temperatures and humidities and reducing oxygen levels. Okay? Okay, let's review what we went over today. Okay? We went over photosynthesis. We went over cellular respiration. We touched upon cellular respiration. And we did fermentation. Okay? Okay, that's all we have time for today. Next time we'll go again through cellular respiration, glycolysis, the Krebs cycle, electron transport. We'll also go through secondary metabolites, and we'll also go through digestion and assimilation. Okay? So if before then you need to ask me any questions, please feel free to call me or email me or come see me in my office hour, of course, anytime. All right? So hopefully this material isn't too overwhelming. I know that we covered a lot, okay, but hopefully the more we review, the better we'll get it. That's why we went over it several times, photosynthesis, and we'll go through respiration at least one more time. Okay? Okay, so until next time, that's it. Okay, we'll see you. Bye-bye. Aloha.